Gene therapy. What is gene therapy? Gene therapy is an innovative approach that uses therapeutic gene to treat, prevent, and potentially cure a disease or a medical disorder. It seeks to modify the gene expression or alters the biological properties of living cells for therapeutic use instead of using conventional treatments like medication and surgery. Genes are the blueprints, providing instructions on how to make proteins for the body. When a damaged or mutated gene produces a missing or faulty protein, a healthy copy of that gene is delivered to the cells inside the body through gene therapy to restore the normal function of that protein. Human gene therapy is a promising treatment option for a range of diseases that are unresponsive to other therapies. Examples include inherited conditions, viral infections, and certain types of cancers. The goal of gene therapy is to reverse damage caused by any faulty genes that you may have, and fixes a genetic problem at its source. Nevertheless, further studies are still required to ensure the safety and effectiveness of this technique. DNA delivery into cells can be accomplished by multiple methods. Commonly used viral vectors in gene therapy are retrovirus, adenovirus, adeno-associated virus, herpes simplex virus, lentivirus. Non-viral vectors like liposomes, injection of naked DNA, polymers, peptide DNA complexes. Physical methods include microinjection, electroporation, gene gun, synoperation, magnetofection. Chemical methods include oligonucleotides, dendrimers. Let's dive into the details on the methods of gene therapy. Therapeutic genes can be administered through in vivo and ex vivo delivery methods. In vivo refers to the process of direct delivery of functional copy of a gene into the patient's body either systemically or locally to a specific organ using a vector to treat a mutated or missing gene. Common organ targets include eye, brain, liver, skeletal muscle. Ex vivo refers to the process of removing specific cells from a patient's body, genetically altering them in a laboratory, and then transplanting these modified cells back into patient's body using a vector. Common cell types include stem cells, B cells, T cells. For any type of gene therapy to work, genetic material first needs to get inside the cells of the diseased person. This can happen in any one of the two ways. In vivo gene therapy on the left starts with packaging the therapeutic gene into either a viral or non-viral vector. Viral vector carrying the therapeutic gene is locally injected to the specific organ inside the patient's body. Systemic infusion is done in case of non-viral delivery system. When the treatment is performed ex vivo, the cells are initially extracted from the patient's body and cultured in vitro. Viral or non-viral vector expressing a therapeutic gene is transfected into the cultured cells. Transduced cells are selected and amplified in vitro. These genetically engineered cells expressing the therapeutic gene are transplanted back into the patient body, usually by an intravenous infusion. Now, let's take a look at different gene therapy products. As we describe gene therapy, we often say genetic material is delivered to cells to correct the underlying genetic problem that causes a disease. Genetic material is a broad term, which commonly refers to DNA or RNA. There are two main types of gene therapy products, namely DNA therapy and RNA therapy. DNA therapy is the use of DNA to treat a disorder. DNA plasmids. These large, double-stranded circular DNA molecules are genetically modified to carry therapeutic genes into the nucleus of human cell to treat a disorder. Viral vectors. Therapeutic genes are packaged inside the capsid, which is the protein shell of the virus. These modified viruses are used as vectors to deliver healthy genes to the nucleus of the human cell. Viral vectors are safe to use because all the infectious viral genes are removed, and thereafter, they lose their ability to cause infections. Bacterial vectors. Genetically modified bacteria are used as vehicles to carry therapeutic genes into human tissues. RNA therapy. It is the use of shorter sequences of RNA to treat or prevent a disease. There are many different types of RNA therapies. mRNA therapy miRNA therapy, siRNA therapy, suppressor tRNA therapy, ASO therapy, RNA aptamer therapy. These RNA therapies are delivered using either viral or non-viral vectors, and needs redosing to maintain therapeutic benefit, since the DNA is not altered or supplemented. Effective approaches to clinical gene therapy include gene addition or augmentation, gene silencing or suppression, gene editing, gene transfer to specific cell line, which is categorized into somatic gene therapy abbreviated as SGT, and germline gene therapy, abbreviated as GGT. Gene addition or augmentation. The addition or substitution of genes with loss-of-function defects is called gene addition. This is a cell with loss-of-function mutation. Now, a functional copy of a missing or faulty gene is inserted into this cell using a vector. The newly transferred transgene is present in the cell nucleus together with the defective gene. This is the corrected cell. Gene silencing or suppression. The suppression of gene expression at transcriptional or translational levels is called gene silencing. A different approach is required when the disease is caused by gain-of-function defects. Gene suppression is achieved by the import of inhibitory sequences into the target cells. The expression of disease-causing gene is downregulated, and subsequently, the amount of a specific protein made in a cell is decreased. Gene editing. It is a highly precise technology where the genetic material is delivered directly to edit parts of DNA located within a cell to correct the gene defects. It involves adding, removing, or altering specific elements within the affected individual's gene. Gene editing uses CRISPR-Cas9 technology to cut at specific spot in the damaged DNA. It leads to permanent genetic changes and reverse the disease processes. Genome editing can be performed both in vivo and ex vivo. This cell has a defective gene. The enzyme nuclease induces site-specific break in double-stranded DNA, which stimulates the two repair mechanisms, with or without the DNA donor repair template. Now, the gene defect is repaired using either homology-directed repair or non-homologous end joining. In HDR mechanism, the DNA donor repair template is engineered into the cell to correct a mutation, and in NHEJ mechanism, random insertions and deletions are introduced in the repaired sequence. Functional loss of a gene leads to protein knockdown. In germline gene editing, if the modified gene is delivered into reproductive cell, all cells, such as sperm cell, ovum, early embryo, muscle cell, blood cell, and nerve cell undergo gene editing. This manipulated gene is also passed on to future generations. In somatic gene editing, if the modified gene is delivered into target somatic cell, only target cell, like muscle cell, or blood cell, or nerve cell undergo gene editing. This manipulated gene is not passed on to future generations. Another gene therapy approach is gene transfer to specific cell line. Firstly, somatic gene therapy. It involves the insertion of therapeutic gene into somatic cells excluding the germ cells or gametes. Therapeutic effects are confined only to the treated individual, and are not inherited by future offspring. It is carried out in both animals and humans. This approach raises fewer ethical concerns compared with germline gene therapy. Secondly, germline gene therapy. It involves the delivery of edited gene inside the germ cells or gametes, that is sperm or ova. During reproduction, these gametes fuse to form a zygote, which thereby divide, and the modified genes are passed into all other body cells during the development of offspring. It is heritable as it alters the genome of future generations. It is very costly and ethically controversial. It is performed in animals, but legally prohibited in humans in various countries such as US, UK, Switzerland, Australia, Germany, and many other countries around the world due to unknown long-term risks in future generations. Let's list out few potential benefits of gene therapy. Gene therapy can potentially slow down or stop the progression of many diseases, such as heart disease, AIDS, and cancer, and inherited disorders, such as cystic fibrosis that have limited treatment options. Gene therapy may reverse any damage in the patient if early treatment is given in the course of disease. It offers the possibility to design treatments that can specifically target any of the thousands of genes in the body. Gene therapy can target a specific cell type to prevent toxic systemic effects. Decreases the cost of therapy in the long run. Coming to the potential risks of gene therapy. The risks associated with gene therapy depends on the type of therapy and the type of delivery mechanism to the cells. Gene therapies are not a guaranteed cure, cannot ensure beneficial results, and probability of unexpected side effects. Introducing foreign material into the body may trigger a negative immune response which prohibits future participation in clinical trials. Persons are monitored for years after gene therapy because the treatment could produce either good or bad long-term impacts. Sometimes, the modified viruses may regain the capacity to cause disease in the patient. At times, insertional mutagenesis may occur in gene therapy if the virus inadvertently targets the wrong cells. If the DNA is integrated in the wrong place in the genome, for example in a tumor suppressor gene, it could induce a tumor. Cells' genome integrated with therapeutic DNA must remain functional, long-lived, and stable. Many cells divide rapidly, and so the patients ought to undergo gene therapy repetitively. Inserted therapeutic gene may result in unexpected gene expression. Potential treatments in clinical trials and the FDA approval process takes many years. Gene therapy is associated with few ethical concerns. These treatments should be thoroughly tested as they involve the use of altered genes and potentially dangerous viruses. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.